Hello again, everyone. Welcome to Q&A with A&V. I'm Vincent Racaniello, and joining me from Columbia University, Amy Rosenfeld.
Hello, Vincent.
How's it going, Amy? You have a good day?
Let's see. Did I have a good day? Yeah, actually I did. So
Good.
we did some science today, so it's good. Made some slides.
That's good. Simon wants to know if we have any good vaccine jokes, but someone had a virus joke earlier. It was, do you know what a virus has to do to transmit? It has to strain.
Has to strain?
Isn't that
Why
funny?
Does it have to...
Sure. Become a new strain. You get it? You get it,
I don't know why I do get it, but I don't know why it's funny because I don't understand why it needs to become a new strain to transmit. Old strains transmit all the time.
It's taking the rhetoric that's flowing around and making a joke out of it. I really liked it. Liked it.
It's good. You can like it.
Right up front, let's comment on why Brazil rejected Sputnik. It was actually a misunderstanding. And we talked about this in the latest TWIV, which will drop at midnight, but I'll tell you the bottom line. So the adenoviruses that are in the Sputnik vaccine are replication defective because they're missing a gene called E1A. And to propagate those vectors, you grow them in cells that have the E1A gene in them. And sometimes the virus can recombine and pick up the E1A gene from the cell. Very low frequency. It's known to happen. So what you do is you do a plaque assay on cells lacking the E1A gene, and you see if it makes plaques. And so I happen to have the document from Russia. I'm going to show it to you. Oops. Here it is. You see it there, Amy? Can you see?
Yeah, I see.
So this is the result of a plaque assay where they tested different batches of the vaccine. Here are the dilutions and the number of plaques. You see 0, 0, 0. And the positive control, you can see they're getting adenovirus plaques. So the best you can say here is there's less than 50 plaque-forming units per dose of vaccine. That's because you can't go to a lower dilution. But there's no virus here, and the Brazilian authorities didn't like this less than 50 per dose, and so they rejected it. But Amy knows that that's the limit of detection, right, Amy?
Of course.
So the vaccine is fine. It was just a mistake.
You forgot my name, they're saying. You didn't put my name there. I'm nameless.
Sorry.
It's okay.
Got it. I need to be corrected.
Well, it says Amy doesn't have a handle, so we can't. So can we speak directly to her? I don't know what that means.
Well, I'll get Amy.
Name is missing.
Next week, I'll get Amy a YouTube account, and she can answer. You guys can talk to her directly, and
I need a YouTube account.
Yeah, but that'll just give you a handle.
Okay.
Thank you, John, for your super chat. You liked the joke. It wasn't mine. It was someone else's who I should give credit to. I don't know if I can find it.
Well, maybe
it's already gone. But anyway, you could look it in the chat history. All right, let's see. Let's get some. And by the way, folks, if you put at Vincent Racaniello in front of your question, that'll save us some time because I know you guys are chatting here. That's great. Chat amongst yourselves. And if you want to ask us a question, at Vincent Racaniello. And a number of people are asking about the situation in India. Will it make a difference in the world? Yes, of course. You know, the viruses don't respect borders. And so I'm sure the outbreak will spread to neighboring countries, which are just as crowded as India and have not great healthcare systems.
You mean viruses don't have passports?
Do they have vaccine passports, Amy?
I don't care about a vaccine passport, but how are they going to get stamps to know what countries they've been to? It's kind of depressing.
I want to, by the way, announce that we now have two mods. We have Frank P. And thank you, Frank. And now we have Vanity Nutrition.
Who?
Vanity Nutrition. This is
a new
Vanity
listener?
That's Vanity Nutrition. She was
Good,
actually a
thank
student
you.
in my virology course, and I recruited her. I said, you should know some virology now. So thank you, Vanity, and thank you, Frank P. Frank
Excellent.
P is here as well. Okay, so the reason India is blowing up is because, they locked down last year and then they relaxed and then people went crazy. They had big religious and political gatherings of millions of people and boom, it's not the variant, it's just SARS-CoV-2 and people behaving
Well, I don't.
badly.
Did you talk to Nisha? Because I don't think he thinks that they had a great lockdown, and I don't think he thinks that it's because they had all these gatherings. I think he thinks it's because they didn't take it seriously. It's overcrowded and took a while to launch. You should talk to Nisha because several of his childhood friends have died. It's been a fatal infection for many of the people that he knows.
Amy, here's one for you. With all the masking, there must be a lot less common cold viruses circulating. What do you say, Amy?
So yes, are we defining common colds as the coronas and rhinovirus and flu?
Paramyxos, adenoviruses, all of them, no?
Yeah, there was a huge decrease in diagnoses. Yes, biofires were quiet. Yes,
But
true.
now they're reemerging, right?
Yes. Well, not all of them. Rhinoviruses have reemerged significantly. The other ones are still depressed. There's not as many different kinds of flu and RSV and parainfluenza and stuff as there are rhinoviruses. I mean, you should know the title of the first slide of my talk. Enteros are the most ubiquitous human pathogen, and that includes rhinos.
That's right.
But what's even greater was that gut pathogens also decreased. You saw very little norovirus and stuff. So it wasn't just respiratory viruses.
Amy, what do we think of the Tafix nasal spray?
I don't know.
Any thoughts?
What is that?
Well, I don't think it's a low pH spray.
No, I don't. I'm not interested. I'm not into it.
It's not really going to be sufficient. It will have a transient effect, so I'm not impressed.
I'm not into it.
Hi, Amy from Houston.
Hello, Abby.
And when we know if we need to get another booster, I got my second Pfizer. Oh, I'm sorry. Amy doesn't like me to say that.
No.
And I'm a teacher in New York.
Yeah.
Elena, pazienza. It will be sometime. I don't think you're going to need a booster this year, maybe next year or the following. What do you think, Amy?
We'll see. It really depends on how long immunity lasts, right? Could be 5 years. Maybe it'll be like tetanus. You need a booster every 10 years. I wouldn't worry about it quite yet.
If someone presented 20 different T cell epitopes, would you expect production of twice as many total T cells as you would get with a similar thing presenting 10 epitopes?
Why would you expect twice as many total T cells?
Well, the thing is, Doug, you already have all the specificities in you. All the T cells you're ever going to make are in you in terms of specificity. If you get infected with something and the T cell doesn't recognize it, that's it. You can't make a new T cell. Those are all made during gestation. So you have them all already. And if one presents 10, it'll amplify those 10. If one presents 20, it'll amplify those 20s. That's how I see it. Lost Laboratory, is that like the incubator? I really like the name Lost Laboratory. If this person didn't have it, I would take it and use it to name.
You're the incubator.
And now we're the incubator. I was just thinking the other day, Lost Laboratory would be a great name. Maybe I can license it from Lost Laboratory. Any word on studies looking at combining different vaccines? The old mix and match. There was one combining Pfizer and AstraZeneca, right, Amy?
Yes.
No word on it. I think people are getting lax about it because, if you come from another place and you got an inactivated vaccine, you could walk into now a supermarket and get whatever you want. Eleanor wants to know if you wear sensible shoes to the lab. She wears Christian Louboutins with the red bottoms.
Not to the lab, I don't.
No, she wears sneakers, right?
Sneakers, loafers, hush puppies. You should always wear more than one shoe per week.
I have 2 pairs that I alternate, but nobody cares about my shoes. Melissa's a psychotherapist in Queens. We're talking about having face-to-face sessions with high-risk patients, some vaccinated, some not. We would be in offices with masked sessions, 45 to 50 minutes. Thoughts? Well, Melissa, if you're vaccinated and masked, you're good. 1 patient at a time, I presume, with you, so you'll be fine. Right, Amy?
Yes.
A good scientist should always cite sources, even for jokes. Yeah, I looked for it. And the person who gave the joke was here in the beginning, and I just lost it. So let me see if I can find. Here we go. I'm in the beginning. Here it is. I'm going to find it before we move on. There's an RNA. Oh, no, that's me. That's me telling a joke. By the way, Amy, I said there's a joke among us. We have an RNA structure called an iris. What color is an iris, Amy?
It's multicolored.
Yeah. I can't find the joke. Damn. Do these things go away?
I don't know. You're in
Well,
charge.
here we go. This is from Whale Oil Beef Hooked. Whale Oil Beef Hooked. He said, or she said, what does a virus need to do in order to reach more people? It needs to strain itself. I love it. That's the original. Love it.
It's good.
Good job, Whale Oil Beef Hooked.
It's good.
Do you know if SARS-1 had variants, Amy? I don't think we knew, right?
Not to my knowledge. There were 8,000 cases, and we didn't do a lot of. It was really before the explosion of high throughput sequencing. So, you can't work on the virus anymore, really. So, although Vinny just has a paper on it.
Well, you could work on it if you're in a select agent lab, right?
Yes.
You need FBI approval.
Yes, I have it.
Amy is FBI cleared, folks. She's the real deal.
I am. I could go and work
so
on
Mark says,
I
believe India variants are already in a number of countries. Yeah, they are. Not surprising. They're in Michigan, for example. That's a state, by the way. It's not a country. Andrew says, Health Canada has approved Pfizer vaccine for 12 and up today. Some provinces will start offering next week. That's great. I think we're going to do that this week, right, Amy?
I think so. At the end of the week, usually they meet on Fridays.
Farhad says, my mother, 71, got Sinopharm. Exactly. Can't you come to the U.S. and get an mRNA vaccine? Absolutely. Absolutely. Many people have asked us that. I don't see why not, right, Amy?
Yeah. I think it's fine to get another type of vaccine. Yeah.
Here's one for Amy. How many days post-infection would a PCR test show up positive? I've heard people can be positive well after the infectious period has passed.
That is true. The RNA does persist in some cases for months. The majority are PCR negative. Generally 5 to 7 days after the onset of symptoms. But 3% will be positive for a very long time. Like Nishai, he was positive for months, but was not shedding virus. It just persists for some reason that we don't understand.
People of concern, exactly. And I have that on my slides.
That's good.
I should show people my slide for tomorrow. Let's see if I had it. No, I don't have it here, unfortunately. But yeah, I always say it's about people of concern. It's not necessarily...
It's about behavior of concern, not
behavior of concern. That's right. People are fine, right?
Well, I don't know that they're fine, but it's behavior.
Well, you're fine, Amy.
Okay, that's good.
Canada has 530 cases per 100,000. Isn't that one of the highest in all of North America? Alberta has a COVID behavior problem. I wouldn't be surprised, right? That is
Not
very high.
surprising.
It is
Yeah.
very high.
But not surprising.
Do you have an idea why vaccinated people in Seychelles are having a new surge? Is it because of the efficacy of Sinopharm? So if that's what they're using, that's about, 50 to 60%. So yeah, there are a lot of people who are not protected, and then probably they haven't vaccinated everyone, right? There's still susceptible people left.
I'm sure.
All right, this is a question I get asked all the time. Maybe Amy can shed some light.
Okay.
What is your take on Geert van den Bosch's views on the vaccines targeting one protein causing selective pressure on the virus to become more infective?
Okay. It's not causing, it causes selective pressure for changes, but they do not result necessarily in the virus becoming more infective. It's a misuse of the term. Term is not used properly. So yes, I would agree that having immune pressure on one protein causes it to evolve maybe quicker than other proteins, but it doesn't result in more infectivity. It just results in different changes.
And
What
I
do you
think,
think?
so as far as we know, only the spike gives rise to neutralizing antibodies. Would you agree with that, Amy?
As far as we know, but that's, I mean, I don't know what we've looked at, but yeah.
So there's not much else that we could have done unless you want to make T-cell epitopes where the pressure wouldn't change them. There wouldn't be selective pressure to change
What
those.
do you mean there's not much we could have done?
To make other than a spike. We could have included other proteins, but they're not going to give rise to neutralizing antibodies. That's my point. So to say we're causing selective pressure by spike-only vaccines is overlooking the fact that for making antibodies, we have no other choice. Now, if you want to make...
That's not what the question is.
I understand, but I'm going beyond the question. I'm going beyond
it. You're... Okay, whatever. I disagree with what you've stated, but that's fine. Because actually what you've stated is not... You will also, if you go back to TWIV and listen to Paul and Theodora, the pandemic explorers, you will see that Paul says exactly what I just said. And you agreed with him.
I agree, but right. I think there's another nuance.
Truth of the matter, no, there's not really a nuance. There's other ways to have made the vaccine that wasn't one protein only. The whole pandemic has been focused only on neutralizing antibodies and solely on spike. You have no idea if they're neutralizing antibodies against anything else, any of the other proteins that are in the membrane, like envelope and membrane.
Yes, agreed.
So
I agree that focusing on spike.
You've overlooked all of this. So the
I think
truth of the matter is,
focusing on spike has put pressure on spike to change, hasn't it?
Of course, that's what I said. That's what Paul said.
I agreed. I didn't disagree with that, but I also added other things, but you didn't like it, so that's fine. Not a problem.
I mean, it was like a bunch of, I don't know, it didn't make any sense.
Are you saying I rambled, Amy?
You rambled in a non-scientific fashion that then just like kind of cut your nose off to spite.
And I rambled and I droned. That's the other thing I did. Okay. Marty said, Marty is in the 3-day genomic conference. I can't believe how much I know thanks to the TWIV team. Well, that's great. It's really good to hear. Oh, this is interesting. Mark says, do you think folks coming back from abroad who received Sinovac will be considered fully vaccinated in the U.S.? Ooh, I haven't seen that question before. I think if they have received the vaccine approved in another country, then yes, they will be considered fully vaccinated. What do you think, Amy?
You have to be.
Yeah.
I mean, are we going to then say that no other country is vaccinating their people?
Yeah, I agree. Pete is about to finish the course. Very good. Good job, Pete. What's an ideal RT or R? What is RT or R? What's a bad RT? I don't know what RT is. You talk about R-naught, reproductive index. I don't know what RT is, but let's say you're talking about reproductive index. The lower, the better. If it's less than 1, you can stop an outbreak. If it's more than 1, you will have an outbreak. And if it's really high, you can infect a lot of people, like measles. So the best is less than 1. 1 or more is not great, and more than 5 is not good at all. As Amy would say, not good at all.
Yes, that is true.
Hit the like button, folks. Who should not watch virology lectures on your channel? Oh, who should not? I don't think there's anyone who should not, Amy. What do you think?
Everyone should.
Everyone should watch them. I would say anyone less than 6 months of age, you don't want them to hurt their eyes with the video.
Can they even focus? Can you focus on that?
I don't know. But I think everyone should do it. Oh, so Mark, you got the AstraZeneca. And I think you'll be fine. And you'll be protected. Don't worry about it.
My friend got the AstraZeneca. He's fine.
In Canada?
Yeah. Andrew went and got the AstraZeneca a couple of weeks ago. It's fine. I think his second dose is at the end of July.
So Credulopathic Remedy asks, what would happen if we give patients lots of Tempress 2 or Cathepsin L to cleave spikes prematurely before they can infect cells, or maybe a custom enzyme that causes premature cleavage? How do you think, Amy?
There are a lot of other targets to these enzymes. I'm not so sure I want my cell proteins cleaved randomly.
Yeah. So
I'm not a big proponent of that.
Yeah, it wouldn't be safe. Okay, Marco says, what can you do to a virus to make it more virulent, pathogenic, and infectious? Well, you have to take a whole virology course to learn that.
That is true. And generally, you don't compare. You don't say one virus is more virulent than the other virus, nor do you say it's more pathogenic than another virus, nor do you say it's more infectious than another virus.
So it's complicated, Marco, because we haven't been able to do this, in fact, so we don't know any generalizations. By the way, I'm jumping down to the chat. Lucky Sixes said, Michael Osterholm should watch your virology lectures. And then someone else said,
Apurva
yes,
should watch.
Apurva should watch too.
Apurva, yes. She should stop writing so much.
Apurva is a writer for the Times. Some of you may know she does a lot of the COVID coverage. And I wrote to her yesterday. I said, look, I am tired of your narrative about transmission and the variants. And she wrote back, she said, well, you're the only one who says they're not more transmissible. So who should I talk to? So I sent her some names.
Susan
and Nels and the guy at UCLA. I forgot his name.
It's good people.
I didn't give your name because I know you don't like to talk to the press, Amy.
Yeah, I'm not that big into talking to the press.
Brian
Because
says,
my question always is these are ridiculous, these are hypothetical, ridiculous conversations. And I want to know how they're going to address the immediate problem of ending the pandemic today or preventing the next pandemic, right? And then they don't respond. Then because they don't they have nothing left to say. So I'm like, okay, well, so when you come up with an answer for those questions, come back to me and we could discuss your question.
All right. So Adam here has some insight into what we were just saying. He says,
wait, where is his thingy? Oh,
I have to find it. Here we go. From Wikipedia, basic reproduction number, R-naught, is not the same as the effective RT. Okay. So R-naught is the reproduction number of a virus with no mitigations. So early in an outbreak, you calculate it based on some isolated transmission chains. But then as you start to do masking and distancing or even vaccination or whatever, then you can calculate it, but it becomes R sub T. It's the effective, because you can change the reproduction number, but not the basic one, which is fundamental to the virus. That's what that is. Sorry, I didn't get that. Thank you, Adam, for the clarification. All right, back to Tony Fauci. He stressed the importance of a second vaccine for variant immunity T cells. What about J&J? I'm not clear on what this means for one-dose vaxes. Are these effects lower? Thank you. What do you think, Amy? So J&J was designed as a one-dose, right?
yes.
So it means that it induces memory and T cells sufficiently in their view, right?
Yes.
What's the
I'm not
sugar?
sure he was thinking clearly.
Tony?
Yes.
Sugar.
It happens.
With a cute cat, what is the latest research on how long one can go between first and second shot? Amy, you sent me a preprint the other day, didn't you?
Yes.
What did it say?
It was the other day, Vincent. I've read 30 papers since then. So, I mean, seriously,
Oh, I know what it was. So there
I
was
sent it to both you and Daniel.
yeah. This is
I've read 30 papers at least since then. Really?
So here it is. So it's a JCI paper. The first dose mRNA vaccination is sufficient to reactivate immunological memory in recovered subjects. So it's not exactly what you're asking, sugar.
But it's close enough.
Close, but the best is a few months ago out of the UK, where the AstraZeneca was delayed, and they showed increasing development after the first dose for up to 12 weeks.
Yeah, I mean, they're doing it 4 months in Montreal, and Andrew is fine. So you can probably delay it probably a year, but people are going to forget to go and get vaccinated because they're going to say the pandemic is over, I don't need it, blah, blah, blah.
Emerald X wants to know if we've looked at a preprint showing maybe spike alone could be causing damage. Did you see this, Amy? It just came out.
Yes, I did. No, it didn't just come out, because this is exactly what we discussed in November when I was on Twiv about the Catania paper, and Alan got all upset when the lady died.
Yeah, that was a patient, but here they're producing spike alone, and it's causing some cell damage. I mean,
they produce spike alone in IPS cardiomyocytes, cause damage, cause the tsunamis fusion of calcium and electrochemical dysregulation, because it caused the tsunamis of calcium. Remember?
Yeah.
And the guy said we misused the word heart attack, and he corrected us and said it was electrical dysfunction, not heart attack. That has a separate, that medically has a separate definition.
It's true that a spike can cause cells to fuse. It can cause damage. Whether or not that's happening regularly in an infection, we just don't know, right? And Emerald wants to know, in the context of mRNA vaccines, would that be a problem?
No, because it's only being produced by the cell that you get that got the uptake of the vaccine, and that's not a cardiomyocyte. Doesn't seem to do anything in the muscle cell of your deltoid. Yeah,
so much misinformation out there. Just have a chat with Amy someday, and she'll tell you about it. I have a chat with her every day about it. Margot says, I read. We didn't talk today, actually, but we texted, and we tend to complain in our texts, right? Margot wants to know.
I was making a list. I needed to make a list.
Margot wants to know about a Pfizer third dose in less than a year. I heard that the Moderna, Amy sent me this, the Moderna modified mRNA vaccine for the South African variant works well. Mm-hmm. I don't know about. So I saw the article from Pfizer. They think they might need a third dose, but I think we have to wait. We can't tell. And anyway, it wouldn't be a big deal because we change flu vaccines every year or so. So it's not a big deal.
Right.
Dr. Crotty made a remark about viruses that are susceptible to neutralizing antibodies. Are there common viruses that evade neutralizing antibodies? Yeah, well, if you have a spike change, for example, if it's a spike-bearing virus, a single amino acid change could make the virus not susceptible to neutralization by an antibody.
But that's not common. That's not a common virus. SARS-CoV-2 is not a common virus. Common viruses are like the common colds. Of course, they evade somewhat neutralizing antibodies.
Yeah.
Otherwise, they would die out.
And as someone asked me this earlier, and I said, well, rhinoviruses exist in over 100 different, what should we call them, genotypes, Amy?
There are over 160 different isolates.
And
Genetic
they don't confer,
isolates.
do they confer cross protection? There's some drift between them, right?
Yeah, there is some drift between them, yes.
So that's the strategy of that virus, to have hundreds of variants circulating at the same time. This is something near and dear to Amy's heart. She's working on this right now.
I am. It's very near and dear to my heart.
Is the vaccine effective against P1? That's the Brazilian. Yes, it is. A number of companies have done trials. Yes. However, when you have many people unvaccinated, the vaccine will not confer protection. And that's what's the problem down there.
Well, it does confer protection, just not to enough people.
Where are we with the transmission studies in college kids? I think they're ongoing, right?
I don't believe it was funded. I believe it was stopped for lack of funding.
Some people are saying you wear Crocs, but that's not true, right?
I would never put a pair of plastic shoes on my feet. Never. Oh my God. No. Not only that, I'm not Mario Batali. No.
He wears orange Crocs, right?
Yeah, that's what he's famous for.
So Overjoyed is doing it at Vincent Racaniello so that I see your questions versus your chatting. How can an individual find out if their immunity is still active post-second vaccine? Well, you could look for, there are two tests. You can get an antibody or a T-cell test. Those are commercially available, right, Amy?
Yes. I mean, I don't know why you would think it's not active post a second vaccine. That's the point of the boost.
Hey, Amy and Vincent, can you explain what antibody titers measured in units per mil mean in terms of concentration? Tracking mine over time for science. Amy's doing a lot of this these days, right, Amy?
I am. So it's not it's an activity. So over time, you're going to need more of your serum to have X amount of activity. Units is a measurement of activity. So it's usually, this is the amount that's required for 50% neutralization. And then when you go to titer it, then you go and you say, when you come back, you do it slightly, you find out that you need to have more serum to have 50% neutralization. It's an activity. It's not concentration.
Yeah, usually you'd make dilutions of serum and you report the highest dilution that gives you neutralization, right? And that's like if it's 1 to 1032, the titer would be 1032. But some companies convert it to units. I don't know what. But
a unit is defined generally as like when you buy interferon. Like when we buy interferon, it's defined as a unit, and a unit is defined as the amount that is required to protect cells from. Think it's 10 to the 3 of VSV. So 10 to the 3 plaque forming units of VSV.
Right, got it.
So an antibody titer is the same thing.
Charlotte says the top virologist in Columbia, that's Columbia University, said there's a new variant in South America. Is it Columbia, the country? No, it's the university.
No, she spells it the university. And who's the top virologist? Ho?
Well, I think that's what they think, but I think you're the top virologist at Columbia, Amy.
I don't know about that.
I don't
I'm
care
just
if there's a new variant.
But I'm not clear what he was talking. First of all, I'm not clear what he's referring to, and I'm not clear why he's saying this. I mean, I have not agreed with many of the things that David has said, like the idea that there's a lack of conserved amino acids or areas that cannot change. There's always areas that cannot change.
I think wherever you look, you can find a local variant. I don't think this is a big deal. We should track them, I agree, to make sure they're not anything unusual, but I don't think this is news.
Even if they are unusual, what are you going to do?
Keep vaccinating.
Well, you're going to keep vaccinating no matter what. So, as I said last week, are you asking a virus evolution question, a scientific evolution question? Or are you asking how to case manage? Because if you're asking how to case manage for patients, it doesn't matter. You're doing exactly the same thing.
We track vaccines in other countries. Do we track therapies and availability? I'm not aware. Are you, Amy?
Not aware, but I do know that somebody in Canada wanted one of their family members to get monoclonal antibody therapy, and they were told that they could not get it because the person was not in the hospital. And it was kind of like, really? Have you not been listening to TWIV? Listen to Daniel. He tells you when it should be done.
Hubert wants us to discuss original antigenic sin and any possible effect on top-up vaccines. What do you mean by top-up? If you get an extra boost or something? Well, if you've had one vaccine, and you're always going to make a memory to that. Top up.
Top off. Well, could you top off my drink? Or would you top off a beer or something?
So I'm not sure, but so far, these viruses are all very similar, so I'm not sure that this is an issue.
I don't believe it's an issue.
Did you see the podcast with Rogan and Rogan? They had an interesting conversation about the origin of COVID-19. No, I didn't, and I'm sorry, I'm not going to watch it because I don't really think they know anything to be discussing that, and I know that will offend some people. Didn't John Oliver trash Rogan recently, I heard, on one of his shows?
I believe that's what you told me. So all of a sudden, these two brothers are evolutionary virologists?
Apparently.
Oh. All this time I wasted 6 years of my life getting a PhD and another 5 doing a postdoc to be considered somewhat of an expert or have some higher level of knowledge than the common schmojo. You mean I could have made a lot of money and been less stressed in my life? Really?
You could have just had a podcast and then you would be an expert.
Oh, damn. I want my money back.
Yeah, I'm going to get Amy another screen. Actually, I have a screen here. I have a laptop with no sound, right? Because you don't want to echo. And I watch the stream on that as well as on my streaming software here. So I'll get Amy that. We'll take care of it. I have an extra laptop for you, Amy. And you are an absolute queen.
I guess that's good. Thank you.
The Moet Chandon. I keep the Moet Chandon in a pretty cabinet. How much sequencing is the U.S. doing? Do you know, Amy?
Not enough.
Yeah, I don't know the number. It costs money. And
Biden just put in $2 billion for sequencing or something. Yeah.
I'm not sure.
When he came into office.
I'm not sure that we need to sequence more. I think we have to have a representative sampling. But as Amy just said, what are we going to do? Amy is frozen. And your image is degrading, Amy.
It happens. I'm old. It's like Miss Haversham from Great Expectations.
How complete is our knowledge about the human virome? Go ahead, Amy.
0.
It's very low, very, very understudied. We should know more.
It's
I
very
agree.
hard, but it's very hard. I mean, people have done it. People only really characterize the microbiome because they took advantage of, 16S, right? And there's no such thing as a 16S in virus. And what was it? Carl Reese, the guy in Illinois or Indiana? Woes, Carl Woes.
Carl Woes, yeah.
Yeah.
So here's another one for you. What's the speed of sequencing? How long to do SARS-CoV-2?
Depends on what you do, how you do it.
So you want to do whole genome?
Could take overnight. Well, it could take overnight if you do it the old-fashioned Sanger sequencing way. It could take a couple of hours if you use like a nanopore or a MySeq. Depends on what you're doing.
What's the fastest? Fastest a couple of hours?
Yeah. I mean, I don't really understand why this matters. Who cares what the speed of sequencing is?
They're just curious, Amy. That's fine. It's fine to be curious.
Yeah, but
by the way, I sequenced polio. I sequenced polio. It took me 1 year in 1980.
Yeah, you have to. Yeah, I know.
I'm eligible for a vaccination in the UK. Can't get one. Less than 30% has had 2 doses, and people are behaving like it's all over. Let me show you this interesting picture. This is the share of people who have received at least 1 dose. Israel is the highest, followed by the UK and the US. And look at down here, India is as low as the world. That's part of the problem. Much of the world has an issue. I think they have another slide here showing it on a world map, yeah. And you can see much of the world doesn't have vaccine. It's not good. Would you agree with that, Amy?
There's weakest as the country that is not vaccinating.
Will the cells that present SARS-CoV-2 antigens from the mRNA vaccine be killed by cytotoxic T lymphocytes?
They can be.
Yes, they can be. Absolutely. Remember to like the video. We would appreciate that. Thank you very much. Can you start saving these on Spotify and Apple Podcasts? I'm not sure that many people would be interested. I've had a few requests. UK, non-COVID respiratory virus surveillance. Low flu, RSV, paraflu, human metapneumo, lowish adeno, but peaks of rhino in the young when schools opened. There you go, Amy.
Well, yeah, that's what I've been saying. And that's what I heard at the AFM meeting last week. Yeah.
Is anyone working on a West Nile vaccine? Well, there's already one for horses because horses are expensive, especially racehorses. But yes, people are working on them for people, and there will be one at some point. Is it bad to give...
It's 8:45. We have 2 more questions.
Oh, boy, the time goes so quickly.
I know, and I'm busy. I have to get back to work. And I think we have mice tomorrow. I have to check.
I thought it was Friday.
It is Friday. Oh, yeah, it is Friday. We're bleeding a lot of mice.
Okay. Is it bad to give incentives like free beer, free tickets to the vaccinated? Will it make people hold out for the goodies? Why is the messaging so bad?
What does messaging mean?
Well, tell people you can get a vaccine if we give you free beer and tickets to a concert and so forth. I don't know if it's bad. I think if you can get people vaccinated, it's good. What do you think, Amy?
I'm fine with it. We give people free things all the time for less important activities. I'm fine with it.
Do PF, the Pfizer drug, and molnupiravir act on different proteases? Well, molnupiravir is an RNA polymerase inhibitor. The Pfizer is a 3C-like protease inhibitor, right, Amy?
Yes.
And this is a one that may come out by the end of the year.
Yes.
I think Florian Kramer said vaccination rates were quite high in the Seychelles, joked about going on vacation there. And you want to go there?
I don't follow what he says.
After AstraZeneca and J&J, are adeno vaccines dead?
I don't believe that they're dead. I think they still have a use, but I would not have phrased the question that way. I would have said, with the efficacy observed using mRNA-derived lipid nanoparticle vaccines, is that technology going to overtake the other ways of vaccinating? And I would say yes.
One more.
Okay, one more.
Charlotte is going to teach an art appreciation junior college, 36 students per class. I'm 78. Students are masked. Vaccine not required, but Charlotte, I'm sure you've been vaccinated. What do you think, Amy?
She's fine.
Enjoy.
I think she's also the woman who suggested that she would make my mobile. But she's fine.
Okay. Yeah. Amy, thank you again.
My pleasure. See you tomorrow. I'm sure we have stuff to do, but I don't know what it is.
See you tomorrow. Have a good evening.
Oh, we have to practice my talk. That's what it is.
Okay.
Practice my talk. All right.
Bye-bye.
Bye.
All right. Stuck with VR. Do I need a name here? Sure. Why not? Thank you for your super chat. Can this virus mutate itself into extinction? I doubt it. No, I don't believe it will. I don't know any examples. Viruses that go away never get started, essentially. But it will not mutate itself into extinction, no. Denise wants to know what would have given rise to T-cell immunity. Well, in what? In infected people, people vaccinated, all the viral proteins, well, not all, many of the viral proteins have T-cell epitopes. Those are short parts of the proteins that are recognized by T cells. And so whether you're vaccinated or infected, and if you just got a spike-bearing vaccine, you would make T cells against those epitopes for sure. I need to get to bed early. I can't stay for the whole chat. Sorry. I've been fully vaccinated, but I feel uncomfortable with what is happening in India. Will we be okay? Yes. If you are vaccinated, you will be okay. There's no indication that those viruses circulating there will evade the vaccines. And you've probably gone to bed. Sorry. Sorry about that. So N doesn't cause neutralizing antibodies. Not that we know of. So here's the thing. I'm going to get a virus for you. Here's my SARS-CoV-2 model, which was a gift from Mark Holger. I forgot the name of your company, Mark. I'm so sorry. Here's the spike, right? And so this virus attaches to cells, and if you have antibodies against the spike, like these yellow guys here, it will block attachment. It will block attachment. The N protein is inside coating the RNA. So even if you had antibodies against it, right, there's no way the antibodies can get to the N protein because the virus is infecting you are not cut open like this, right? So they wouldn't neutralize. So N antibodies will not neutralize. You will make N antibodies for sure because that's the way antibody production works. But they will not neutralize. The base came off here, which has a little antibody on it as well. I'll fix that. Okay. Show and tell. Love it. If you compared the number of PFUs in a very fresh nasopharyngeal swab in transport medium with the same specimen when it arrived at a lab after typical transport time conditions, what would you expect? Well, it depends on how much time. If it's within a 24-hour period and it's been, let's say it's just kept at room temperature, right? No frozen, nothing like that. You would probably lose up to 10 times, a tenfold infectivity, a log of infectivity in that time. And longer, you would lose it all. Infectivity, that is. But you can still detect nucleic acid by PCR, of course. Can rhinos catch rhinoviruses? Well, they're called rhino because the rhino knows, right? Dr. Kapila, an infectious disease expert at Rutgers, contracted COVID after 2 shots of Pfizer in India, died. How common is an escape? Well, the vaccine is 90-something% efficacy in the trial, so not everyone is protected. He could have been one of those unfortunate people, or he may not have responded properly, which happens in a small percentage of people. And of course, these cases are amplified by the news. They're picked up and amplified because the news wants to make a story that the vaccines are not going to work, right? How would one go about identifying other neutralizing antibodies to other proteins like nucleocapsid? Well, what you could do is you could immunize, say, a mouse with virus particles and isolate individual antibodies against the different proteins, the N, the other. There are 2 other proteins in the membrane, this blue one and this white one here. Maybe antibodies to those would be neutralizing. So then you would take those antibodies and do a neutralization test in cell culture. You infect cells with or without antibody, and you look for plaques. And this is what Amy does all the time, every day. And it's really simple to do. Of course, SARS-CoV-2, you need a BSL-3 lab, which is a specialized lab that not all of us have. There is one, 2 at Columbia, and Amy has privileges to work in one of them. Will we need an updated mRNA vaccine booster shot? Pfizer CEO says yes, but it seems like current vaccines work quite well. First of all, the CEO doesn't know much science, okay? I don't mean to insult him, Alex Azar. He's a great business person. He knows about supply chains. He knows about motivating people, and he gets his science from people who tell him what to say. All right? I don't think we know. Right now, his vaccine works against, his company's vaccine works against the variants. How does he know? Does he have a crystal ball? No. He's making guesses, or his people are making guesses. So I say we have to wait and see. Would the vaccine impact the length of time active virus can be detected with PCR? So what you're asking is, if you are vaccinated and then you get infected, will you have a shorter duration of shedding detectable by PCR? Yes, because that's the whole point of being vaccinated, right? Yeah. Are 20 to 60 participants really enough of a phase 3 for the Pfizer for the 12 to 15 to know what the risks are? It's obviously, if there are rare side effects, it is not enough. But this is an emergency, and we're making trade-offs all the time between people getting back to school and being protected. So that's what's being done. I would like more as well. But the risk of severe COVID is lower in the younger ages as well, so you have to factor that into account. It's a good point, though. Vaccine glut in the U.S. is depressing me. Yeah, it's unfortunate that vaccine rates have gone down. We should really be using up all the doses. As I said earlier, I went to Acme the other day, and there's a sign out front. It says, come in and get vaccinated. No appointment necessary. It's free. And other countries in the world, as I showed you in that slide, I show it again. Look at this. Share of people who have received at least 1 dose. I don't want to do that. Many countries have not even got 1. And the darker countries here have a lot. And so we're very fortunate to be in this position, right? And I agree with you. I'm very sad. Yeah, and when you walk into your drug, there's a tech asking if you wanted to be vaccinated. It's really sad. Totally. Columbia said they would consider students vaccinated with vaccines not approved in the U.S. Thank you, George. Because you have to be vaccinated to attend Columbia in the fall, and they should accept vaccines from other countries. Totally agree with that. Wild Bunny, if it were up to me, I'd be sending vaccines all over the place. But that's why I'm not the president of the U.S. My view, I think the world should have one healthcare system that serves everyone. Now, that would be a bear of a healthcare system, but it would ensure equal distribution of tech to everyone. Just like I want to give knowledge to everybody through this, my podcast, videos, this. I think everyone should have knowledge. I think everyone should have healthcare and education. And that's why I'm not a politician. I'd like to have your thoughts on this. Why vaccinate 70%? Shouldn't we just vaccinate who wants it and let the others be? Well, you could, but then you would have outbreaks regularly and people would die, and people who don't want to be infected might die. So I think we should get higher. I think we should get 70%. And then I agree after that, maybe we should take it easy. But we're going to keep pushing because I'm hoping that if the infections continue, that'll be an incentive. So I think now in the U.S., with people not having to wear masks outside, there will be slowly more and more cases, and maybe that'll be an incentive for people to get vaccinated. Yeah, it's really a problem. You want to let the people who get infected. Yeah, but that's not fair. In a situation like this, you put a strain on the healthcare system. You let people die unnecessarily. So I'm not sure that's a good strategy. If I go to the U.S. Open in September, people come from all over the world with the variants, say they're all shedding, vaccinated. You'll be fine. You won't get sick. You might be infected, but that's going to be fine. Your immune response will control the variants. There's no variant except for the South African variant, which I think is hard on many vaccines, although the J&J vaccine did prevent death and hospitalization in South Africa. I think the vaccines will handle all the variants that are around so far. Don't worry about the variants. Absolutely. How, when my family and children masked, did my wife and daughters come down with strep? Well, they may have touched themselves, got contamination on a hand, for example. I don't have any experience across the U.S. and Canada. How could I get into my field in microbiology? Volunteer in a lab. I know you need money, but volunteer for a while. Show that you're eager, and then they'll hire you. How can hospitals improve their HVAC to limit airborne infections? Well, this is a whole science in hospitals that's been studied for years. They have to have, HEPA filters in key places. Far UVC is another way to disinfect areas. This is not my expertise, but hospitals are versed in dealing with this. It's a profession in its own. Why watch a lecture if everyone will die soon? I don't think everyone's going to die soon. Everyone dies eventually, but between now and then, we might as well learn things, no? How effective is the Pfizer vaccine against a B1.617? It depends what you mean by effective. Preventing mild disease, it may not be great. Preventing death and hospitalization, probably 100%. That'd be my guess. As I said, I'm not worried about these variants. They are things that happen. They happen in every outbreak. We're just over-sampling and overreacting to them. And I do think they're as serious as everyone makes them out to be. I do think we should track them for sure. No, you shouldn't retire from teaching. Do you want to do it? That's all that matters. If you love it still, if you have the passion, then do it. Don't let the pandemic interfere. But if you're fed up and you had enough and you want to do other things, yeah, then retire if you can afford it. Once in a lifetime. Why should we vaccinate children since they hardly get sick and the vaccinated can still transmit and get infected? Seems like a waste of vaccines. They hardly get sick, but it's not zero, as Daniel Griffin says, right? And so do you want a kid dying because they're not vaccinated? Maybe your kid. I don't think that would be right. I think we have to be wary of everyone. I'm seriously sick of the misinformation about COVID and the Times. I canceled my subscription. I'm very unhappy with their COVID coverage. I agree with you. I totally agree. And Raphael agrees. The Boy Who Cried Wolf was written 400 years ago, and the media is still selling papers with the same premises crying doom every day. When will humanity learn? Never. Because it's a capitalist economy drives it, and whatever drives it. Yeah, children can get sick. It's not zero. Not zero. If a person is fully vaccinated and contracts COVID, are they protected from serious illness, death, or are they not protected at all? Kind of like you can't be a little pregnant. Wow, this is a different situation. If you are fully vaccinated, so first of all, understand that no vaccine is 100% protective against disease. So with that in mind, you can be protected against death and hospitalization, or the other order, hospitalization and death. And I think it's independent of the variants that are circulating. The variants are not responsible for any particular situation. If there's an outbreak, it's because people are getting together and transmitting. It's not because of a particular variant. So it depends on the vaccine and what percent of efficacy it is, and also where you are. Population matters as well. What do you say to people who don't get vaccinated because long-term effects are unknown? There are no long-term effects of any vaccine that come up after 2 months. There are rare effects like we saw with the J&J and the AstraZeneca, which you don't see until you immunize 1 million people, right? Or more, because you need a few of them to accumulate. But there are no long-term effects that you would pick up after 2 or 3 months. And so they've been fully tested. The only thing that we learned from longer trials, and these phase 3s are continuing for another year, is the durability of the immunity, how long it lasts. That we don't know. But that's not a side effect issue, as your friends are telling you. Renzo Dalla Via, ciao, come stai? Can you please reflect on SARS possibly evolving to be more suited to our species? For example, change at D614G. So what's happening as the virus is moving through the population? It's the first time this virus has been in humans, right? It was in a bat beforehand. And it is mutating as viruses do. And then changes are selected that provide better fitness for the virus. And many of these changes, I think, are driven by antibody selection. And we only saw them at the end of the last year when many, many people had been infected. So those changes are continuing. They're going to continue probably for a long time. It's more suited to our species. I think it's very well suited already when it first came out. And I think the changes we're seeing have to do with antibody pressure. And that's it. It was already very transmissible, didn't have to get better. If there were any changes in transmission, which is very hard to nail down, they would be minor, I think, because remember last year it was already spreading very well before any of these changes arose. In the long term, I think what will happen is most of the population will become immune and there'll be far less death as a consequence. And I think it may become another common cold coronavirus. Yeah, BBC breaking news. The South African variant is in East London. Well, you know what? It was probably there weeks ago or months ago. Not an issue. Not an issue. Vaccine will still work. Get vaccinated. Maybe that's the idea. Maybe the press is got something up their sleeve and they want to scare people to get vaccinated by saying the variants are going to get you. I doubt it. How does common cold get around T cells? No viruses typically. Well, it depends on when the T cell response occurs. For example, influenza virus, the T cell response is essential in your recovery from infection. And that T cell response kills cells in your respiratory tract. That's why it takes a long time to recover from flu. I'm sure T cells play a role in common cold as well. Now, some viruses can get around them. They have a variety of strategies for evading T cell killing, which we don't have time to go into. But they don't work perfectly for every virus, but for many they do. Are there updates as clinical studies stating effects? They're still spreaders. There have been a few small preprints out suggesting that transmission is depressed, but after vaccination. But the problem is that these are happening very soon after vaccination when antibody levels are still high. And, after vaccination, antibody levels decline very rapidly. And so I think that people will be infected, but I suspect that the memory response will dampen transmission. I'm not too worried about that. I think most viral vaccines do prevent transmission. That's why herd immunity works, right? My guess is that vaccination rates will continue to fall in the U.S. till there's a large proportion of the population that are unvaccinated, keeping it going. I think that might be right, although it may be more of an epidemic scenario. However, the rest of the world, remember, many countries are still not vaccinated. They probably won't be for many years. So this could go on for a while. However, look, the 1918 flu ended in 4 years without any interventions. And we're doing interventions now, so this has to end sooner than that. I hear that the people in New Jersey are getting their third shot of tequila. I wasn't aware. However, I've been in my basement all day, so I don't know. I've been here lecturing and podcasting and making presentations. Manolo Blahnik, red soled pumps, yeah. Yeah, you talk to Amy, you hear about shoes, for sure. One of the authors of Spike Alone Damage said experiment used amounts of intact spike vastly above that expected to circulate. That's correct. Often these experiments are done in unnatural situations or conditions, right? Can you start this week in cancer? I would like to, actually. I'm not an expert, but I could get some cancer experts. I would love it. That would be fun. Because when I go to the studio, I'm going to be doing a lot of recording. I'm going to be uninterrupted. So speaking of my studio, any updates? So it was our goal to move in May 1st, and obviously we missed that. The place we wanted on 8th Avenue, they never responded to our offer. So we went last week and looked at a new space on 7th Avenue. It's a block south of Penn Station, nicer space, and we're making an offer on that, and hopefully June 1st we can move in. But I will get pictures and videos once we get the space. I have to go back Friday and tell them where I want the walls moved and put up and so forth. Oh, someone else thinks Crocs suck. Yeah, well, they're just plastic shoes. Your thoughts on the adeno, which caused thrombotic events? Well, these seem to be vaccine associated. They're rather rare, and in the U.S., they happened in women in a certain age bracket. So in the U.S., the decision was made. It's up to you whether you want to take this vaccine or not. Discuss it with your doctor. So if it were me, I'm outside the range. I'm a male, and I'm outside the age range where this happened, so I would take it. So I think the events are rare. And in Europe, they decided that the risk of COVID outweighed that. And it can be up to you as well whether to take the vaccine. I think that's probably the way to do it. Why the vectors cause this is very interesting, and I need to really look into that to get some clues, because as you know, some adenoviruses can bind red blood cells, and I just wonder if that has anything to do with it. But then you would see thrombotic events in adenovirus infections in humans, and I'm not sure we see that, or if we even look for it. Doctor in India says they're giving steroids and ivermectin. He said they know it's not effective, and Merck said it was not effective. Right. So why are they using it? Do no harm is what Daniel Griffin says, right? Christian Louboutin. Yeah, what did you say before? Oh, yeah, it was Christian Louboutin, not Manolo Blahnik. But that flows off my tongue too, doesn't it? Would I be able to get both doses of Pfizer and a single dose of J&J? Okay, so yes, you could, especially as things are loosening up in terms of appointments and so forth. The only thing is they may ask you if you've been vaccinated, and you have to decide what you're going to tell them. I think it's not harmful at all. What's all the nonsense about shedding from vaccinated individuals causing illness and menstrual irregularities? How did this get traction? Mainstream media, unfortunately, amplifies. You're probably talking about the school in Florida that said this. I don't know where this comes from. Nobody asks me, but you guys do here, so, and Amy as well. People with G6PD get vaccinated. I don't believe there's a restriction there, according to Daniel Griffin. I think he said that. Colombia, South America. Sorry, we were railing against David Ho, and you meant the country. Oh, boy. Michael Osterholm says, fully vaccinated should party hard and have people over, but stay when going out. Okay. Michael Osterholm, who said March would be the worst month ever in the U.S. That's the problem you get from predicting, right? Oh, arthritic flare-ups post-COVID. Yes, I have heard that too, because, you know, the vaccine, or you're talking about COVID, the disease, causes inflammation, and so that's going to play into arthritic flare-ups. I'm not surprised at that, for sure. Oh, would the mRNA vaccine, the spike, damage more cells if you put it into the bloodstream? I think not. You know why? Because when you put things in the bloodstream, they go through the liver and they get filtered out pretty quickly. So we know that from experiments with viruses in animals. When you put viruses into the vein, they get filtered out really quickly. So I think that would not be an issue. Not going to be an issue. Can a virus-infected cell produce antibodies? Not typically, no. The cells that produce antibodies are B cells, and some viruses can infect B cells, actually. Epstein-Barr virus can infect B cells and immortalize them, make them memory B cells. But, you know, COVID, as far as we know, doesn't infect B cells. Chris, 3-day tent revival, 200 people. I am expecting COVID will be one of the guests. I'm fully vaccinated. Should I worry? No. If you're fully vaccinated, well, look, again, no vaccine is 100%. You have to take your chances. You know that the Pfizer and Moderna are over 90%. The J&J is pretty high. I would wear a mask if I were you. I still wear a mask outside because, well, I'm actually in the habit, and I have to go into buildings. I park my car and walk into Columbia. What's the point of taking it off and putting it back on? I have to wear it in the building as well. Virus breakthrough. Are those people who do not mount sufficient immunity no matter what? Well, it depends. No, not no matter what. Some people have genetic diseases that prevent them from mounting immune responses. So that would be no matter what. But other people just randomly don't respond to a particular virus or bacterium. We don't understand why. So it's not all or none. Do we know if immunity wanes in babies after mom stops breastfeeding them? Yes, eventually it will. If this is a kind of passive immunization, right, where you're getting antibodies from someone. So eventually they'll go away. It's like monoclonal therapy. You get a shot, an IV or an intramuscular inoculation. It lasts a few months, and then it's gone. Same thing with passive antibodies from your mother. Yeah, we do call it Pfizer. It was invented by BioNTech. Yep, I agree, JM. It's easier to say Pfizer. But you're right, it was invented by BioNTech, and Pfizer just took it. But they do have a big marketing machine, right? Yep. I get myself in trouble, yeah, for sure. Let me look for some, and Amy takes care of it. Yeah, it's fine. Yeah, we can have huge arguments during the day. It would be great if I could invite, the founders of BioNTech, sure, but they wouldn't come to TWIV, this tiny little podcast, would they? I guess I could try, but they have PR people that are shielding them. I don't want to deal with that. I just don't want to deal with that. Yes, Charlotte, if you get COVID, you can request monoclonal therapy. You should. But I don't, and it could be that you're in the percent that are not protected. So yes, you would get monoclonal. You definitely should get them. Yeah, I know, Molly. It's not an argument. I was just saying that. However, Joe Rogan doesn't know anything about virus evolution. That was Amy's point. Why should I listen to him, waste my time when I could be explaining things to people? I'm the one with 40 years of virology experience, and he's not. He's a great podcaster in what he does. It's fine. He has millions of followers, fine, and I have not so many. But I do a narrow thing. And look at Cosmos. Rogan said he was a comedian, not to take stuff he says seriously. He says things to get listens. So anyway, this virus came from nature, not from a lab. Yes, BAM is a single monoclonal, so it does not neutralize certain variants. And so you have to take cocktails of monoclonals now. You shouldn't take a single monoclonal. Did Amy say home, Joe? I didn't even hear that. Could you be downplaying the possibility of lab origin because it could result in lower future funding? No, I don't think so. I'm just looking at the science, Uncle Billy. I have a good friend who's Uncle Billy. It's not going to impact funding at all. But I think the data says it came from nature. That's all I go by, with all due respect. As far as I understand, all our available vaccines target antibody production for the spike. Well, they give antibodies, but they also give T cells. Okay, Karen, you're forgetting that. They make T cell against the spike epitopes, and that's very important because that protects you against disease and death. We could target other parts of the virus. Yes, we could add an mRNA for N protein to get more T cell epitopes, for example. And I think next generation vaccines will see that probably, most likely. I don't know what Rogan talked about, and people are saying I didn't address it. Fine. Anything he's saying about lab origin is wrong. He doesn't understand it. And I don't have time to go into it because we've talked about it over and over again. And it's a waste of time. And the only thing that will put it to rest is to find the source in nature, as we did for SARS-1. Why didn't people say SARS-1 was made in the lab? Well, there wasn't social media back then as much anyway. And we found it within a couple of years. What is it? Maximum Gilbert gels. That's Tom. That's how I sequenced polio. It took me a year to do that. Yep. Yes, Australia has a very low vaccination rate. That's what I showed you here. Look at that gray. Well, it's no data. So you might be right. The Seychelles is the highest. Let's look and see. This is the little island here, right? Or maybe it's, I don't even know where the Seychelles is. I'm showing my ignorance. Sorry, folks. Super chat from Credulopathic. Thank you. Do you know if the intellectual property for the particle vaccine from Reed will be put in the public domain? If it's from Walter Reed, it should be, right? If that's the only entity involved there. And now I feel like a jerk because there's an island in the Indian Ocean at the eastern edge of the Somali Sea. Wow. It's really in the middle of nowhere. Okay. It was close here. It wasn't so far away, folks. It's like up here. It's up here north of Madagascar. Right. I pointed to India, but I was wrong. It's around here. Can't see it on there. And it doesn't show up on this graph. We put all our eggs in the AstraZeneca basket. We did. And in countries where it's used, it seems to be doing well. Thank you for the super chat. Post-polio news studio update. We did that, I believe. A lot of good questions here, folks. Don't know how to get through them any more quickly. My gosh, it looks like I need another hour, right? Thank you, T. Klepper, for your super chat. By the way, all this donations go towards supporting the studio. And someone said last week, it's going to take a lot of super chats, but it's not going to all be supported by super chats. We have other sources as well. The mouse isn't showing up. Sorry. Well, I can't. So here's how I get the mouse to show up. I share my screen and then I have to go to my computer and move it. Okay. And I just forget to do that. I'm very sorry. But anyway, here the Seychelles are up here. That little dot. And I don't know what the color is. That's the problem there. And unfortunately, the comments come so far after I make the mistake. It's horrible. Why is the COVID in India more infectious? It's not more infectious, folks. It's just a bigger wave of infections. Well, I think if I blew up the map, I could see it. Carlos, you're welcome. Were you in my course? You came to office hours, or is this office hours? This could be office hours too, right? Antibody-dependent enhancement risk with vaccine. No evidence for it at all. I haven't seen it. None of them reported. So I don't think it's an issue. So I know that I haven't addressed Rogan. Some of you don't like that. I'm sorry, but I don't want to do that. In Australia, we have only access to AstraZeneca, 55 and up. Orders for Pfizer and Novavax not likely to the end of the year. I'm sorry to hear that. Let me take a look at if that was on the map here. No, Australia wasn't on the map of one-dose vaccines, unfortunately. Can the body develop resistance against adenoviruses? Yes, they can. So a third boost of an ad vaccine would be difficult. But with mRNA vaccines, obviously, it's not an issue. I know, I should ask. I'm limited by time. I do everything for all this myself. I don't have anybody helping me, so some things slide because I don't have a chance to do that. In the beginning of the pandemic, scientists expected many vaccines to fail. Any idea why so many are good? I don't really know. I thought the mRNA vaccines would not work because I couldn't imagine putting mRNA into you, but they did a good job of protecting it. It's brilliant development. So that's two. And the adenovirus vectors, well, we knew they worked. I guess we didn't know that spike would work, but they work with varying efficacies. So I can't say why they're so good, but, I'll take it, right? Why not? Media issues. Cranks will pounce on anything they think supports them. Deniers pounced on Fauci's dead nucleotides remark on Twiv. Yeah. I did a interview with Peter Daszak back in December 2019 in Singapore at a NEPA meeting. And people are pulling that out. They're cutting a piece of it out where he talks about what they were doing in one of the labs in Wuhan and saying, look, this proves that they engineered this virus. And it's just crazy because they hear what they want to hear, right? Will, any data on the Lucira tests? No, I've heard that they are 30 minutes. I don't know anything about the accuracy. I'm sure the data are out there, but I haven't looked at it. eastern edge of western Massachusetts. Eileen, I think you've been here before because I sort of remember the eastern edge of western Massachusetts, unless you wrote in a letter to TWIV. Let's see. Tom, thank you for your super chat. Any amount is appreciated. Sean, thank you for your super sticker. So I use a streaming software called Ecamm where I can highlight the chats as they come in. But if it were in the YouTube chat, you would see this pair character bowing down saying thank you. But, if you go to the real chat, which you guys are in, and I can see it on my laptop where I am following the chat. See, a pear-shaped character bowing down from Sean. Do you think there's a difference in glycosylation of Novavax's spike because it's made in insect cells that may disrupt efficacy against B1351 relative to Pfizer? I think that's an interesting possibility. I don't know that it's been explored, right, in terms of where the epitopes are for antibodies. That's an interesting point because, yeah, there was a little less efficacy there. In Australia, 0.62% have received first dose. That is really, really low. Hey, Eileen, see, I remembered. It rang a bell, the eastern edge of western Massachusetts. Love it. Now we're at the end of the chat, so it's easier to answer you in real time. Forlos comments says, oops, that's the wrong person. mRNA vaccines activate a huge portion of T follicular helper cells relative to other vaccine modalities. Is that true? And if so, why is that? Do we know? You're welcome, Ditching the Grind 3. It's my pleasure to be here. 535 of you have been here. That to me is just amazing that you would come here and have this little community. So we're going to try and build some communities elsewhere as well. We're going to build a Discord server. We're going to build, well, I'm not going to build it. I'm going to have one. I'm going to have more regular chats, Ask Me Anythings, that is, weekly, to try and spread my information as much as we can, and mine and my colleagues' information. Does Australia need help? Well, it seems like they do because they're waiting for Pfizer and Novavax, right? It seems like it's going to come, but not till the end of the year. BAM works for the UK. Yeah, it's some of the other variants are not working, and you need to use a cocktail. But if you don't know what's infecting the patient, you give a cocktail, right? Okay, thanks for that information. That's really interesting about the T follicular cells. And Wendy, thank you for your super chat. Waiting for the shoe to drop in New Zealand. Weird vaccine rollout. You'll be joining that Discord. Great. We'll be publicizing it as soon as it's set up. It's going to be a community thing because I want to build a community, not just mine, right? If someone had an unusual reaction to the vaccine, would it be safe to assume that COVID would cause the same reaction? No, not necessarily. There's two different things. Very different things are happening in vaccination versus COVID. So, no, not at all. Hello, Rodney from McLeod, California. And Sandy, thank you for your super sticker. And here, it's a pear character under a rain of confetti taking his hat off to say you're amazing. And looking, you can see that in the chat, but not on my chat here. Any new relevant studies about natural infection immunity? There was another one last week saying that it can last up to a year. So that's great. I'm sure it will last longer. John Sardella, you're welcome. It's my pleasure, and thank you for coming. It's amazing that you guys are coming here. It's the composition of the LNP that determines whether the vaccine is tolerogenic versus immunogenic. That's cool. And you got the same RNA inside it, right? What's the light bulb on the desk? Oh, that's very cool. Let me show you. This is a glass virus sculpture. Unfortunately, I don't have autofocus on my camera, so I'll put it next to my head. This is by, who's the guy who does the glass sculpture in the UK? I forgot. Anyway, my co-workers bought this for me. My co-podcast hosts bought it for me on our 600th TWIV last year. This is a very expensive glass sculpture. It's called Future Mutation. It has spikes, and inside you can see a genome. You know, he made this before the pandemic. What the hell is his name? Come on, folks. You guys must know who it is. Anyway, it's sitting on a light, see, so that it glows. And I'm thinking of moving it closer because you can't see it, right? It should be here. What the heck is his name? Glass virus sculptor. Can't be many of those. Luke Jerram. Okay, that's what that is. And thank you. I did show this a while ago, and I guess there are a lot of new people here. That's so cool. Isn't it beautiful? It's thousands of dollars, I think. I don't want to drop it. It's glass. And it arrived in a big crate from the UK, and it was only one spike had broken off. One last question here. Is, in your opinion, for aspiring virologists, are genetic and biochem imperative? Absolutely. Genetics and biochemistry. And one more, Luke Jerram, one more. I've been trying to learn more virology. Is there a course more specific to coronas? No, but I could make one. You know, I'm going to do some live streaming courses, and that could be one of them. What do you think of that? I could do it. I can't resist answering questions. Any reports of replicating vector? So we covered this at the top of the show. It's a misinterpretation by Brazil. There's no replicating vector in the vaccine. Okay, folks, another awesome live stream. Great questions. Come back next Wednesday. We'll do the same thing. I usually show up about a half hour early and just chat by text. So you can show up then and get some questions answered. But in the meantime, thank you for joining me, and we'll see you next time. Bye-bye and be safe.